All right. So now going more in depth of the validation protocol. So as you're going, now we're going to the sterilization procedure. So we'll go in and discuss the cycle types, cycle parameters, equipment needed. You need to, before you go into your PQ, which you're in now, you have to successfully pass your IQ, OQ. You need to write down the equipment ID number, calibration date, and then the load configuration, number and location of each item. So we're going into more depth within the protocol, defining the cycle type parameters, equipment needed, the load configuration, how it's loaded, the location of each item, so that what we're validating to is going to be able to be used as we go into production. So more so into the continuation of the validation protocol. So location of biological indicators or BIs, data logger probes in worst case locations in the center, near but not at the bottom. So your data logger probes or temperature monitoring probes. Uh, so you want to capture data as far as temperature, which was identified as a critical parameter. You want to put it in your worst case locations within the autoclave. So areas where you know that temperature more than likely or is least likely to be consistent. You probably uh, will notice that maybe in the center, maybe near the bottom, are your worst case locations. You want to put some um, data loggers to be able to monitor what's going on through your validation in your worst case locations. You want to use at least 10 biological indicators, which is recommended per cycle, and this is a step-by-step listing of how to carry out the test. Then you want to go into your acceptance criteria, list the required results or range of results. So we identified temperature to be one of our critical process parameters. So we need to have a temperature range where we want to stick to. We need to define that, and then we need to say that, hey, this is what our target is. This is our range, and we need to be able to meet that through our validation protocol as we're testing. If we meet it, we pass. If we don't meet it, we got to figure out what's going on and try to fix it, and then retest. Some instances you might have to increase, decrease the temperature range, but you have to have justified evidence as to why you're doing that. You just can't do it. So going into your validation protocol, as you're getting results, you want to record those results. A diagram showing location of all load items within the chamber, items containing data log and BIs, precise location number of these within each item. You want to have your autoclave cycle tape printout. So any of your printouts that contain data, you want to make sure that that's included in your protocol so that you're able to see the data and also recording the data to show that you're meeting any acceptance criteria you set forth. Your BI results and your data logger results are all you're capturing all your data and putting that in your protocol. So now we're going to verification. So as you're getting these results, your results are reviewed for compliance with acceptance criteria. So ultimately, maybe your quality assurance, your engineering specialist, or your manufacturing, they're reviewing the results you have and making sure that it meets what they are expecting as well. And it's also signed by the individuals who execute the protocol and approved that by the validation team. So there's a lot of verification that goes on and signing from different parties within different groups to ensure that it meets manufacturing, meets quality, meets engineering's expectations of what you would need to do to verify or to validate an autoclave. So along in your validation protocol, you want to list the responsibilities. So you want to identify key members or validation team members and their functions. So these are experts or individuals who are assisting with the validation, whether it be drafting a protocol, approving it, or executing it. You want to list the team members and their functions. You want to list your reference documents used to execute the protocol. So any procedures, any guidelines, anything that you're using to help you execute, you want to put that in your reference section so that you're able to go back and know what documents were used to be able to help you validate the protocol. And then revalidation. of course, things don't always go right, so you can revalidate. So revalidation, it can be addressed via continuous monitoring, using change control and risk assessment. So if you need to revalidate, you have to issue after it's validated. You want to make a change. You have to issue a change control and vet why you're making the change, whether it be you want to reconfigure your validation load, or you want to increase or decrease the temperature parameters you set forth. You have to issue a change control, and then you have to assess is the change you're making, is there a risk ultimately to the process, and that needs to be vetted out before you can actually revalidate and make the changes you want to address. So deviations and investigations. So any deviations from the validation protocol during the validation study should be evaluated for impact to study, be documented and investigated. So as I was saying earlier, you're doing your validation, of course, things are not always going to go to plan, but you need to make sure it's documented, and you should be able to investigate any issues that you had, um, and be able to figure out how you can fix it and going and back to revalidate. So if you have any deviations, they should be closed before the completion of the validation activity. So if you have any investigations over before I close my investigations should be closed. If I have to revalidate or if I have to redo things, they need to be done before you can close the validation activity. Um, so a separate deviation form is required for each deviation. So as you have deviations, let's say you have 5, you can't put all of them in one form. Each deviation is handled its own separate unique ID number, and they're listed within the protocol as far as in the deviation section. So you want to that's more of a traceability and be able to exactly know the number and be able to trace it back, the deviations back to a particular protocol and be able to know that, okay, this issue was identified and you investigated it and it was closed. Uh, these deviations should be signed and dated by the person who conducts the investigations of a deviation. That's basic GMP. So deviations should be signed and dated by the person who conducts the investigation, and the report must be reviewed and approved by the approvers of the protocol. So as you have your deviation, you're doing your report and you're ultimately closing it out. It must be reviewed and approved by the approvers of the protocol, because they're essentially are the experts and they're the ones knowing what's going on. And so you want to make sure they're in the loop of the deviations, that it being closed out before the validation stuff can be closed. So after you doing your protocol, you've executed, if you had any deviations have been investigated, they're closed. And then next you have to do is a report. So your report is based on the outcome of this validation study. A report should be prepared and all raw data attached. The report should be reviewed by concerned departments and approved by quality assurance. And report approval shows that the validation was completed successfully and according to the validation protocol. So you have your validation protocol. Once you close that out, then you have to do a report summarizing all results, all your raw data is attached, and that has to be reviewed and approved, including quality assurance. And then once the report is closed, that ultimately closes your validation. So just because your validation protocol is approved and closed, that does not close your validation. You have to have a separate report. Some companies, as you're going through your IQ, OQPQ, I've worked for companies where you have a report for all phases. I've also worked at another company where at the IQ stage, if your protocol is none, that's it. You don't need a report. You only need a report for your OQ and your PQ. So each company has their own rules and standards, and you just have to go based off their guidelines. But definitely, as your OQ, PQ, or actually through it all, expect to have a report, unless each company has a different stance on how they do the validation. And that is the end of the makings of a validation protocol.